Good morning. Today we are going to discuss two topics. First one is the immunization types, and another one is the cold chain mechanism. What is immunization? It is defined as the procedure by which the body is prepared to fight against the specific disease. Help body to fight against specific disease. Mainly two types is the first one passive and another one is the active. Passive and active. Okay, we'll see definition. It is a process of conferring increased resistance to infection. It is a process of conferring increased resistance to infection. It help to get resistance against the infection. Okay. Mainly we will discuss here. First one is the passive immunization. Do you remember in first slide already we have mentioned mainly two type of immunization method is the first one is a passive and another one is a active. Here we are going to discuss passive. First, what is passive? Passive is won't make any challenging to the immune system of the body. It done by administration of serum or gamma globulin from person who is already immunized, affected by that same disease, to the non-immune person. It is transmitted from whom? Immunized person to non-immunized person. Mainly two types. The first one is the natural method and another one is the artificial passive immunization. We will see first one passive natural immunization. This is commonly the immunization is transmitted to mother before and after birth. From whom? Mother before and after birth. Before birth, how it will be transmitted? Through the placenta, and after birth, breast milk. So there is a first one passive natural immunization. Here the immunity is transmitted to the mother to child. First is that birth time, that immunity will transmit it to the mother to child is through the placenta, and after that through the breast milk. Next one is a passive artificial immunization. It is developed by injecting previously prepared antibodies using serum from human or animals. It is injecting previously prepared antibodies. Example, tetanus, measles, etc. Okay, next one is a active immunization. Active immunization or immunity is acquired by activating immune system of the body. We need to underline this point. Activating immune system of the body. Activating immune system of the body. What will happen here? It will develop resistance against disease by producing antibodies following the exposure to the antigen. Here what will happen? Producing antibodies following the exposure to the antigen. Like that the body will help to develop, or body will help to resist against the infections. Like that this immunization will be prepared the body. If suppose that whenever that foreign body will be attacked in body, help the body to fight against. So that is the body develop resist against the disease by producing antibodies following the exposure to the antigen. That is an active immunization. Here actually what they will do, they will give this small dose of the actual that disease pathogens. So that time what body will do, body will develop the antibodies against them. Antigen antibody the body will produce, and whenever the real infection will come, the body cell memory will be active, and it will produce the full antigen antibody, and it will fully cover up, and full phagocytosis will happen during the time of the infection. So like that this active immunization will be help to develop lifelong immunity against this infection. Which immunization we are done against that infection, the body will develop lifelong immunity. Okay, that is a active. So self body develop this immunity against any of the infection. Like that, this active immunization will be active. Then another one is a active natural immunization. The naturally occurred active immunity involved the activation of immune system of body to produce antibodies. It both clinical and subclinical infection. Here what they will do, in whenever one infection already happened, our body will store that all antigen antibody systems. So what will happen next time if that infection will come, that our body our cell memory will tell, yeah, that antigen or that foreign body is coming to attack us, so be alert. So whenever next time that infection will come, the body will be prevent that infection attack. So that is the active natural immunization. Chickenpox. Commonly, if chickenpox affects one person, if he is fully recovered, the next time commonly he will survive if that same community area also if it exposed, because that same will happen because cell memory is there already. That is active natural immunization. And another one is active artificial immunization. Here what they will do, they are administering that same disease condition vaccines or toxins. That time that antigen antibody system will be active, and our memory cells memory will be stored that everything. Whenever that real infection will come, the body will fight against. Phagocytosis will complete, and we will be fully protected. That is active. Okay, then we'll see what is the difference between active and passive. Active immunization can be achieved through the natural infection by microorganism, or it can be acquired artificially through the administration of vaccines. Two type we can do. One is a naturally will our body will be developed, or artificially. So anyway, the body will be developed on capacity to fight against the any of the infections or foreign bodies. That is a active immunization. The antibodies produced in response to the infection. The natural measles virus antibodies produced to response to the vaccine. It may be live, inactivated, toxic. Individual make their own antibodies the same only. And another one is a passive immunity. Here what will it will happen? The individual gain antibodies from another who has produced them. Here the antibody from the another who have produced. The transfer maternal antibodies through the placenta or breast milk. The another person or that immunized person's antibodies transmit to us. That is a passive immunization. Passive. Active is self body is developing, and passive means transmitted from the some of them. Okay, that administration of antibodies collected from actively immune human or animals. That example is varicella zoster immunoglobulin. Okay, then another one we'll see another one term. What is vaccine? Vaccine is a substance that is introduced in the body to prevent the disease produced by the certain pathogens. Vaccine is a substance that is introduced in the body. One thing you should remember, sometime exam university may ask differentiate between active and passive. That simply active is self body developing. It may be we have past experience of infection, or through the immunization our body self developing immunity against that particular disease. That is active. Passive is transmitting antibody is transmitting immunoglobulin A, mainly IgA is transmitting one immunized person to non-immunized person. Example is mother to child. Okay, so next vaccine, it is introduced body into the prevent the disease produced by certain pathogens. So we will see what is the types of vaccine. First one is the live attenuated vaccine. These vaccines contain modified strains of a pathogen that have weakened but are able to multiply within the body and remain antigenic enough to induce strong immune response. What is live activated vaccine? It is a live pathogens that is a weak. That pathogen is not strong, it is weak, but able to multiply in our body, but it won't happen toxic, that remain antigenic enough to induce strong immune. What will happen whenever this pathogen will enter our body will develop against the antigen antibody. So that is the basic concept of live attenuated vaccine. There is a different example, we can say oral polio vaccine, yellow fever vaccine, etc. And another one, heterologous vaccine. It is a subgroup of live attenuated vaccines. Here the pathogenic in the animals but not in human. Here produced from strains that are pathogenic in the animals. It is a vaccine that is confer protective immunity against a pathogen that share cross-reacting antigen with the microorganism in the vaccine. Example, cowpox vaccines that protect against the smallpox in the human. What will happen here? This pathogen is taken from who? Not human. It taken from the animals. That will introduce in body. That one of the example, cowpox vaccine. That cowpox virus, if introduced in our body, it will be protect against the smallpox. It's already eradicated smallpox. So like that vaccine will call the heterologous. It is also one type of live attenuated. The strain or pathogen is taken from whom? Animal. Okay. Then another one, killed inactivated vaccine. This is a death vaccine or inactivated by the chemical treatment or heat. Simple, no? That bacteria virus, it is killed or inactivated. How it killed or inactivated? By a chemical treatment or heat. Example, hepatitis A, rabies vaccine, pertussis vaccine, polio, etc. Then another one, subunit vaccine. Here microbes or subunit vaccines include only the antigen that best stimulate the immune system. Here antigen, subunit of antigen is introducing. So in some cases this vaccine use epitopes, the very specific part of antigen that antibodies or T cell recognize and bind to it. Whenever this vaccine is introduced, this T cells, you know what is the function of T cell? It will help the immunity, so it will bind it, and commonly phagocytosis will happen. So whenever the next time the real infection will happen, that time also this same process will happen. So that is a subunit vaccines. Next one, DNA vaccine. Here the microbes antigens are introduced in the body. Some cells will take up that DNA. Microbes antigens are introduced into the body. Some cells which take up the DNA. The DNA then instructed those cells to make antigen molecules. Some of them DNA vaccine will be introduced. Some of them DNA vaccine will be introduced. That DNA will be instructed to body to develop against that immunity. Or some of them pathogens DNA will be introduced. Now this corona vaccines, I think in Oxford developed like this DNA vaccines, because actually they are taken the DNA of the COVID and they are introducing that DNA in the another vaccine, and it will be introduced in our body. Not fully that COVID-19, corona virus in our body. They are taken only the DNA of that corona. You can check it in newspaper and site also. So that is a DNA vaccine. DNA will be introduced. And another one, recombinant vector vaccines. It is an experimental vaccine similar to DNA vaccine. It is similar to the DNA vaccine. It is attenuated virus or bacterium introduce the microbial DNA to cell in the body. The vector refer to the virus or bacterium used as a carrier. Here what we'll do, here the vector direct one virus or bacterium is introduced in our body. Virus or bacterium. Virus or bacteria introduced in the body. Okay. Then another one is a toxoid vaccines. The time will come to know they are introducing the small toxins introduced, maybe diphtheria, tetanus, tetanus toxoid, small toxoid introduced in body. Then another one, gene deleted vaccine. These are the genetically engineered vaccine which involve the removal or mutation of virulence of gene of the pathogen. Some of them pathogens, the gene will make some changes or mutation will do, that will introduce in body. And another one, peptide vaccine. These are the chemical synthesis of the immunogenic peptides. It will be introduced in the body. Okay, so these are the common type. I will repeat one more time. First one is a live attenuated vaccine, and another one is a heterologous vaccine. Then another one is the killed inactivated vaccine. Then subunit vaccine, DNA vaccine. Sixth one, recombinant vector vaccine. Then next one, toxoid. Then another one, gene deleted vaccine, and last one, peptide vaccine. So if it possible, please identify at least one or two example for each of the vaccines. These vaccines mainly 9 types are, so please identify or recognize at least one example for each of the vaccine. That is your assignment. Okay, so next, these are the common example. Other than this, only here we are not mention all of them. So vaccines type is a commonly live and attenuated name is a inactivated toxoid subunit vaccines, which all of them here mentioned. But the gene related peptide vaccines, DNA vaccines other than corona, so recombinant, there is no example here mentioned. So please identify some of the example regarding these vaccines. Okay, then route, commonly route, one is a deep subcutaneous or intramuscular, most of vaccine, oral for the BCG, intradermal route BCG, when BCG will be administered at birth. Okay, BCG. Then 6th, 10, 14 pentavalent with the vitamin A. Nowadays government is begin the rota vaccine also, that 6th and 14th. 6 plus polio vaccine also government is nowadays introduced. So 6 at birth BCG, vitamin A and hepatitis A. 6th, 10, 14 pentavalent plus IPV, inactivated polio vaccine in the 6th and 14 week, and 6th, 10, 14 week rotavirus pentavalent, and after that 18 week MR. Then another that 1.5 year MMR, then 5th year DT, 10th year TT, and 15 year TT. And one more thing is 3 to 5 year, in between every 6 months we need to introduce vitamin A orally. Okay, that is a common vaccination schedule for the government of India, but in private sector there is some of the changes is okay. So we are going to discuss another one topic. This may ask in short question, or one time I think 5 mark also they are discussed, the university asked. So what is cold chain? Cold chain is a system transporting and storing vaccine at recommended temperature from the point of manufacture to the point of use, where that vaccine will produce through them up to the client, or the transporting or storing vaccine at the recommended temperature from the point of manufacture to the point of use. Point of manufacture to the point of use, point of manufacture to the point of use. So first, who is manufacturer, then distributor, then vaccine depots, provider's office, and client. These are the some of the cold chain importance. I am going to leave. These are the common setup we need. Cold and chain storage equipment. First one, vac-in cold room, then deep freezers, ice-lined refrigerator. Vac-in cold rooms, deep freezers, and ice-lined refrigerator. Vac-in cold room, deep freezer, ice-lined. You can see the diagram. Vac in cold rooms, this is stored up to 3 months. Then deep freezer, here temperatures in between 15 to 25 minus. It commonly used in our PHS and district hospital level. Then Iceland refrigerator, it is common in CHC, PHC also, plus 2 degree to 8 degree. So how much degree we need to keep commonly vaccine? That is 2 to 8 degree commonly we need to keep the vaccine. That's why this Iceland refrigerator is good for keep the vaccine in at least 2 to 3 months. Then vaccine stability. Some of the vaccine stability sensitive to the heat and some of them to the cold. These are the example. Sensitive to heat, BCG, varicella, MMR, meningitis, hepatitis, DT, etc. And sensitive to cold, hepatitis B combination, DT, influenza, meningitis, MMR, varicella, BCG, etc. Okay. Then light sensitive vaccines, BCG, MMR, varicella, meningococcal, and DTP. Then vaccine storage, no food at the medical specimen. Do not place the fridge in direct sunlight exposure area. Do not store vaccine in more than 1 month at PHC. Do not store vaccine in fridge door or solid plastic trays. Keep vaccine away from the fridge walls and cold air events. And use dedicated vaccine fridge. Safeguard electricity supply. No more than 50% full vaccine. Place vaccine is clearly labeled. Defrost or calibrate fridge regularly. Group of vaccine by type. These are the common vaccine storage system we need to remember. So cold chain is nothing but, what is cold chain? Cold chain is nothing but the vaccine is transported from manufacture area to the up to users. Okay, the transporting equipment, how we can transmit the vaccine. First one, cold box, another one, vaccine carriers and day carriers. These are the diagrams. Cold box, and another one is a vaccine carrier. There is a display sir in vaccine carrier, 60 to 20 while we can store, and day carrier, this is a common in sub-centers and PHC. They are using 6 to 8 while we can keep with the ice packs. Okay, so here I am going to conclude. We have discussed two topics. One is the vaccination and what is vaccine. Vaccination is nothing but the introducing vaccine into the body. Mainly two types. One is the active and another is the passive. Active is nothing but introducing vaccine. It may be live or death vaccines is introducing in the body. And in passive vaccination is nothing but that vaccine immunity is transmitted from the immunized person to non-immunized person. Okay, then vaccines, there is a different, mainly 9 types are vaccines. Then another one is the immunization schedule at birth, BCG, OPV, vitamin A and hepatitis also. Then 6, then 14 pentavalent, it will help to prevent against 5 killer disease. And with that, nowadays 6th and 14 week introduce IPV plus 6th and 14 rotavirus. Then another one is a 18th week MR nowadays introduced. Before it is MMR, now it is MR. Then after that we are introducing rubella vaccine also is there in MR, measles and rubella. And another one 5th year DT, then 10-year TT, 15th year TT, then 3 to 5 year in between 6 month we need to introduce vitamin A orally. Then another one more point, pregnant woman at early pregnancy and after 1 month TT will be introduced. Okay. Then another point, what is the cold chain and how it can be transmitted. How it can be transported. First one, cold chain is nothing but the transporting vaccine from the manufacturer to the up to usage of the persons or that client. That is a cold chain system. It's mainly different method we can use, different way we can storage the vaccine. But one thing is keep the electricity is properly in steady. If electricity loss, that vaccine's stability will lose and it may chance to damage, and it won't keep vaccine more than 1 month is good. So some of the system we can keep vaccine up to 3 months. Then main commonly that vaccine carrier boxes is available. That is first one, cold box, vaccine carriers and day carriers. Okay, thank you, have a nice day.